Hello, and thank you very much for the introduction there. I appreciate everybody staying around to hear what we're doing today, because what we're doing is something that really affects all of us. I mean, the problem of antimicrobial resistance or antibiotic resistance or superbugs is a problem that's really caught up on our generation particularly. I mean, as an example, before we get into the presentation, I remember the days where I'd go to the doctor even 10 years ago and they'd give you one prescription of antibiotics and they'd say, take this, problem solved. Now you go to the doctor and they give you a prescription with many follow-on repeats. And the reason they do that is because the antibiotics that used to work in the past with one prescription no longer work. You need to keep taking those antibiotics to hopefully get the same result. And that's just in recent times, us as a generation seeing superbugs evolve, traditional antibiotics no longer work, and it's a time for change. So that's really where Recce is positioned. Usual disclaimer. What's Recce about? Recce is an ASX-listed small-cap stock. We have a whole new class of synthetic antibiotic, and we're a broad-spectrum antibiotic at that, meaning we kill all types of bacteria. Broadly, bacteria are either gram-positive, gram-negative or superbugs, and as you'll see further in, we kill all and we keep on killing with repeated use, which is the important part. So we are targeting with our lead compound the #1 most expensive condition treated in US hospitals, and that's sepsis, septicemia or blood poisoning. We have been advancing our lead compound through the regulatory process. We found our lead compound long ago. We don't do research for research's sake here. It's about bringing it to market as expediently as possible. In a recent submission to the US FDA, we were actually awarded a very prestigious legal status. We got awarded a QIDP status, which stands for a Qualified Infectious Disease Status, and that gives us 10 years of market exclusivity plus expedited review for the life of the regulatory process. So it's all well and good having patents, as you'll see further in, but that government-backed market exclusivity, that's pretty important and very, very unique to us. We're manufacturing our product. I've got it here, which we'll talk about a little bit more in a moment. It's difficult to see from where you are, but that's it in a pharmaceutical-grade glass vessel. Essentially liquid gold. I can tell you there's no bacteria growing in that bottle, and ready to go into humans. So we'll talk a little bit more about that further in. Broadly, this is the capital snapshot. Top 5 shareholders: Dr. Graham Melrose, who's inventor of the technology; Ford Asset Management, an institutional investor out of Singapore. I'm in there at number 3, Michelle at number 4, and number 5, State One Equities. Don't look at that as, oh, the directors are pretty good at issuing themselves stock. I can tell you for certain that the directors are good at buying stock. The directors have personally invested from day dot in developing this product. So we've got a lot of skin in the game, and we truly believe in what it represents. And in more recent times, as we've seen with our QIDP status, the international community is starting to support that as well. 16 million market cap, usual snapshot there. This, I think, really encapsulates what we're about. So 3-minute video here.
There have been no new classes of antibiotics on the market for over 30 years. Recce 327 has the potential to break that mold. Recce 327 is a synthetic polymer designed to target bacteria that cause infectious disease, including ones that are resistant to current antibiotics, the superbugs. Bacterial infections of the blood are deadly and affect an estimated 30 million people worldwide every year. This is where Recce 327 is designed to act. Recce 327 is designed to be administered through intravenous injection, so it can quickly spread through the bloodstream. Recce 327 is designed to adhere to the outer cell wall of the infectious bacteria through hydrophobic interactions. This weakens the cell wall, and internal pressures from within the cell cause the bacteria to burst. The body's own mechanisms can then clear the infection. Normal non-bacterial cells remain intact as they do not contain high internal pressures. Tests have shown Recce 327 is fast-acting, and as a broad-spectrum antibiotic, is effective against gram-positive and gram-negative bacteria, including antibiotic-resistant superbugs. Following regulatory requirements for new antibiotics, safety and efficacy tests will continue to urgently explore Recce 327's ability to address the global threat posed by superbugs. Together, we can make antibiotic resistance history. Visit recce.com.au to find out more.
So broadly, we are a technology. We're not a single product. But as a technology, we have to, for regulatory purposes anyway, focus on a particular market area. And as I mentioned earlier, our key focus or key indication is for sepsis, septicemia or blood poisoning. As an idea of how big a problem that is, it's the #1 most expensive condition treated in US hospitals, double that of the second most expensive, and kills more people than prostate cancer, breast cancer and HIV/AIDS combined. So the market is enormous. It's growing at about 8 to 13% per annum, and there's currently no drugs specifically for treating the problem of sepsis. As we come across to the next slide and demonstrating the problem that we had in the video there earlier, traditional antibiotics are basically a specific fit. They're a singular indication. If you can picture having a bacteria and an antibiotic, when that bacteria mutates, much like a lock and key, the two no longer connect. Now, with a broad spectrum antibiotic, not reliant on what's found in nature, not reliant on what's dug up from the bottom of the oceans, found in the depths of a cave, we begin with the end in mind, and we've designed a totally unique mechanism of action in our antibiotic to tackle this problem of bacterial mutation. So, by example, instead of a couple of active sites, which traditional antibiotics have, we have thousands of active sites. And that means that bacteria, or bacteria in general, can keep mutating as much as they like. We still adhere, we are firstly still attracted to the outer membrane, we still adhere to it, and we still cause that bacteria to burst. So potentially antibiotic resistance through that unique mechanism could be a thing of the past, and that's something we're seeking to pursue. Here's an example of it working. This is a nice one against a staph infection and MRSA infection. Here you have 3 groups of mice. 10 out of 10 with ours survived, 9 out of 10 with oxacillin survived, and obviously not a great outcome without treatment. You look at that and say, oh, that's not so bad, 9 out of 10. Well, that's 100 in every 1,000 patients not surviving. Not a good clinical outcome. But what's most important here is the reason we chose oxacillin is prior to doing the study, we knew the bacteria was a staph bacteria. Therefore, we knew oxacillin was the best antibiotic to tackle that problem. In a clinical context, you don't know what bacteria that patient has. Every hour left untreated, the likelihood of survival decreases by 6%, and diagnostics aren't fast enough to determine what type of bacteria that patient has. So it's essentially guesswork. What we see here, the bacteria happened to be a staph, could have been an E. coli, could have been a Pseudomonas aeruginosa. There's well over 100 different types of bacteria, but we killed that bacteria and we led to 10 of 10 mice in this case surviving, so just one of many tests. That's a curative example. Here we come to a preventative example. What I really like about this one is that you think about clinical use, particularly surgical operations. Here we have an example where the blue mice already had Recce antibiotic flowing through their blood. The top ones did not. And when the bacteria was introduced to the blood, because of the protection mechanism of Recce antibiotic, imagine in a surgical roll, the drip bag that patient has on the surgical table and post-surgery, we did not allow the bacterial count to rapidly multiply. The reason you see the bacterial count falling from time 12 with the other one is not because the body's managing to get over the bacterial problem, it's actually because the bacteria is shifting from the blood into the kidneys, leading to organ failure, terrible result. You don't want that in a human patient. So that's actually a very good preventative example of getting over the problem of an infection before it even exists. Here's another example. This one's in our patents, and this one, of course, like the others just before, were presented to the FDA and accepted. What you see here is you've got the standard bacteria and you've got their superbug forms. What you're looking at there, and you can see the very different and broad types of bacteria, we kill the standard bacteria with the same concentration as we kill the superbug bacteria as well, meaning we kill staph in their regular or superbug form with exactly the same ease. It doesn't matter how much it mutates, it's still bacteria to us, therefore we still kill it. So that's very demonstrative of that broad spectrum approach and that broad spectrum capability. Here's another one. This is one of the more important ones, commercial antibiotic amongst friends, that's amoxicillin, very, very successful drug, about 10 billion US in sales per annum. Here we do a repeatability test. We test against Staph, E. coli, Pseudomonas. After 8 repetitions of amoxicillin against Staph, it no longer worked. 2, E. coli no longer worked. After 25, actually many more, but we'll say 25 for this purpose, we kept on working, and we kept on working with the same ease. And again, that is reinforcing our broad spectrum capability. Here is how it works. Keeping it simple, it is attracted to the outer cell wall of the protein. It's a hydrophobic attraction to the proteinaceous layer of that bacterial membrane. We adhere to that outer membrane. The bacteria themselves, which is unique to bacteria, have a very high internal pressure, causing them to burst. Healthy cells don't have that. Any bacteria in the blood is bad bacteria. And yeah, keeping it simple, we can discuss the depth of that one later. This is what I really like. This is for the first time. We do it in animals all the time, small species, large species. You see them unwell, you see them survive, and you go, what on earth happened there? This is for the first time at a molecular level, you can actually see the product working. And what we have here is very buoyant bacterial cells. After 20 minutes of exposure, you can actually see the bacteria bursting. You can see that unique mechanism of action taking place. And after 180 minutes, it would have happened earlier, the protocol was just designed that way. You've got little more than a splatter of bacteria left on the page, and in obviously an animal or a human potentially in future cases, you would have that mopped up by the healthy blood cells that are running around the body. They love that stuff. So what we've done is we've essentially got to the right to the tail end of our preclinical studies. We do preclinical studies for the purpose of doing it to FDA protocol, to then presenting that data to the FDA, which we recently did, and that went very well. We essentially, you assess efficacy and you assess safety. Safety, 1, does it kill the patient? 2, does it kill the bacteria? Now, if you don't kill the patient and you do kill the bacteria, you've got a pretty good hope ahead. So there's many details there, but essentially one of the favourite ones I like in there is we don't cause mutagenicity or healthy cells to mutate, aka we don't cause cancer. All independently done, we don't have the capabilities in-house, and we do it to FDA protocol. QIDP status, yes, we've talked a little bit about that. There's only 13 drug candidates around the world that have a QIDP status. To my knowledge, we are the only broad spectrum drug that has that, and we are the only synthetic drug that has that, and I think we are actually the only one for the largest bacterial application of sepsis. So that's a very, very prestigious legal status awarded by the US FDA. Patents, beautiful range of patents that gives us a market exclusivity over our manufacturing and both a curative and a preventative role. They're granted many countries. Manufacturing, yes, we're producing this in-house, wholly owned, no licenses to anyone. Producing that in about an hour and a quarter, 99-point-something% yield, very economical, ready for human use, which we're just about to start. Board and management, Dr. Graham Melrose is the inventor of this technology. He's ex-head of research for Johnson & Johnson. I'm sure many of us have heard of that. He was head of research and a director of their board for Australasia of about a decade. And he is a qualified chemist by trade, and he is the inventor of the technology. All the usual people. The one I really like is Dr. John Prendergast. Dr. John Prendergast is a non-executive director. He recently joined the board. He is a chairman of a New York Stock Exchange healthcare company, US-based, very representative of our future and our aspirations ahead. And you know, so often I see these things and they go, oh yeah, he got paid a big lot of shares and a whole lot of cash, $50,000, tiny parcel of shares. He sees the future of what this company may represent, and that makes me warm on the inside anyway. So I like John being a part of the board. Economics of antibiotic development. The business model of antibiotics is failing, and the drug developers are constantly pulling out. And keeping it simple, the reason that is they come up with a new drug, the bacteria mutates, the drug no longer works, they don't get a return on investment. Terrible. If you've got a drug that is not susceptible to bacterial mutation. When you first use it and after repeated use, you potentially have the catalytic key to change the challenged business model that is, and change the antibiotic industry as we know it. And be assured pharmaceutical companies are taking notice of that. US legislative environment, Right to Try Act in the USA, that's a very exciting one. Perhaps we can talk about that later. We're a technology. We've got, rather than just a single product, we've got multiple other market opportunities with our manufacturing process. We can not only vary the molecular weight of the compound, but vary the length of the compound itself, meaning rather than just an intravascular, it could be a topical, it could be an oral. We've got animal data to support that, and we've got patents to back that up as well. So fallback positions, but also market opportunities ahead there, and that's where we are in our status. Overview, I think we've broadly talked about much of that. As an investment summary, basically we've got a whole new class of antibiotic, hasn't been one in over 30 years. We've got the US FDA supporting us through legal statuses to motivate our ongoing development. We've got very warm tailwinds around the world to motivate ongoing development and advancing this lead compound, and the multiple products that leverage off the technology that is, because when you have a manufacturing established for one product, expanding it across another just by changing the molecular weights, etc., opens up new opportunities. You leverage on a lot of the other preclinical data. Basically, you get to open up all of these other market opportunities without starting from scratch. So that's very valuable, and I think you can read the rest for yourselves. So welcome any questions anytime, and thank you very much, everybody, for listening.